Good morning. I'd also like to thank the NHGRI for inviting me and for all of you for these interesting conversations. So about a year ago, I left academia to go into pharma, and I did that just because there's large numbers of genomes becoming available, and I'm not sure how familiar you are with that. So I thought I'd start off with the landscape that AbbVie is working underneath right now, which is we're in the process of sequencing 50,000 Irishmen. 10,000 of those genomes are in-house. Those are for specific diseases. UK Biobank is part of a consortium of other pre-compete pharma companies where there's 500,000 of those genomes working with Regeneron. There's 100,000 of those genomes already done. The next 100,000 will be in March. So we'll have 500,000 genomes sitting there. And then FinGen, there's 92,000 genomes that have been, again, it's another pre-compete that's been done. So AbbVie will have at least 1 million genomes sitting on the table within the next 3 years. And this is happening with a variety of other pharma companies. So I think as we're looking at this, and then we're talking about phenotyping, pharma is also doing very detailed, in-depth phenotyping. So if you look at our clinical trials, we often spend tens of thousands of dollars phenotyping these individuals. So there's incredibly deep phenotyping information that's sitting there. But there's a common series of problems that we're all going to have, which is what do you do with all the data, and how do you do something with that data? And as a community, I think there's some opportunities here that we could be looking at to leverage the data across the different resources. And again, like I said yesterday, with the UK Biobank, we're getting a 1-year lead before all that data is being made public. And so I think there is a framework for being able to work with pharma in pre-compete models. So some activities that I think are incredibly important is how do we come up with some pre-compete projects with this community? So if we really look at this from a standpoint of we really need to understand what genes do. So I think there's places where there could be incredible synergy with this community and looking at a variety of frameworks. And so we're interested, we have this problem that everybody else has, which is there's 50% of the genes we don't know what they do. So we have LD blocks, we have all the population data, we're able to drill in deep, but we still have this fundamental problem of what do you do? What do the genes do? So Joe asked yesterday, well, why should NHGRI be the ones that solves this problem? So I think what's unique about this community is that many of us work in different disease areas, but in some senses disease agnostic. And the disease areas are focusing on that specific disease. And this community actually crosses many different areas. And that framework of thinking about this, I think, is a huge advantage of being able to go forward on this. So how do we leverage that type of information? Computational biology, I think, is a major goal that we need to be looking at. I'm going to make a prediction that 5 years from now, hypotheses are going to be generated by the computational biologists, and the wet lab folks are going to be doing the testing. There's going to be a flip in how we're actually doing this. As I look at the data that we have inside pharma, there is so much data right now that it's impossible for a single human to sit down and sort that data and turn it into something. We can now cherry pick and show anything we want. You pick your favorite gene, and I'll tell you that it can do something important. So we're now at a point where whatever you want to prove, we can prove. This is really now moving to the next level, which is how do we take that information? And again, I think this community has a very unique perspective that can be beneficial around that. So we need more people trained in this area. We need investments into tools and visualizations, as has been mentioned by others. The end game is the clinical phenotype from our perspective. And one of the things that's been interesting in the conversations is phenotyping. And I was thinking yesterday in the conversation around phenotyping that maybe we need to think about phenotyping as what's the question. Because broad-based, expensive phenotyping isn't going to get us there because we can't afford to do it. So how do we take advantage of data that does exist and use real-world data and clinical data? We can all complain about the clinical data. It's messy, but there's millions of those records. We can all agree that we need to have a better electronic medical record. But the fact is there's millions and millions of data points and millions of ways to do this. And so I think eMERGE as being one of the projects that started thinking about this, but how do you twist on that? How do you turn on that and saying, we have this data. How do we leverage this data? And there's some important distinctions that we have to pay attention to. So when we're talking about some of the diseases, the phenotypes that we're talking about, I'll use blood pressure as an example. It's a balloon. We put a balloon on somebody's arm, and we're trying to find the molecular mechanism of that, okay? Many of the phenotypes that we measure, it's very analogous to that. But we're not going to change that that's the phenotype. So we need to be thinking about what that phenotype is, and then what do we need to drill into that. And sometimes I think we get lost in trying to find what the variant is, yet alone what the gene is. If we can figure out what the gene is, then we can start figuring out what variation does. So the whole idea of looking at phenotype versus mechanism is a little bit of that conversation previously. A mechanism is we really know how all this works. That's an incredibly complex activity. Understanding the phenotype, and then we have some associations and correlations with that that gives you a handle to grab a hold of to start working on that is, again, incredibly important. And when we think about this, all these LD blocks are a massive problem. So we, as a huge population cohorts, you can go into and you say, yeah, there's an association. But if you really go in and look at the genes underneath that, I think Eric said yesterday, if you pick your favorite transcription factor in that area, and you think it's looking at that gene, well, there's other data that's sitting there that says the gene next door. Or worse, we've actually gone in and worked up a few of these areas. It's more than one gene sitting in that LD block that are all contributing to that. So these are activities that I think this community needs to be looking at and can address. And finally, the clinical goals, I think I'm going to urge that this community starts changing how we use language around variants of uncertain significance, or VUS. A researcher often talks about it as it's a variant we don't know what it does. So that scares people. There's millions of variants that we don't know what they do. But what Leah talked about yesterday, which was a clinical variant of uncertain significance, is a very different meaning. And when we say variant of uncertain significance as a researcher, that's not the same as a variant of uncertain significance on the clinical side. And I think we have to be careful with that language because we're confusing a lot of people. That clinical director who's written in that report what a VUS is not saying that we don't know anything about what the variant is. What they're saying is that we've looked at this variation in the context of that clinical case and we're not sure what it means. Okay, those are two very different things. So I think the talk yesterday that Leah presented was insightful. Right? So here's the ACMG 59 genes that everybody thinks should be looked at, and there's 23,000 variants of uncertain significance inside this activity. So I think a major goal could be is, can we take on, as a community, 25% of the most important disease-causing genes? Now we can debate what does that mean, but really start going in and looking at it. So if we start at a high level of gene, what does the function do? And then drill down into a handful of those in terms of figuring out what the variants do. And the last piece I'll make is I think this is maybe more for the colleges and the physician groups, but there needs to be some type of evidence codes developed around the model organism database or information that helps drive a clinical decision. So what you may or may not be aware of is that you can go to your zebrafish, you can do your fly, Hugo's having some success, but the vast majority of physicians are not going to use that information, even though you can show the mechanism that's associated with this. And I've done this many times in physician communities. They're not going to make a call on that because there's no clinical trial that proves that. Now, a rare disease gets you around that, and so I think as we look at some of these rare diseases and Daniel's potential project, how do we find these variants that cause catastrophic disease in families and do that characterization and then understand how do we turn around and use that information, I think is another way to think about that. So I'll finish by saying I think the community that's here has access to massive amounts of data, but there's lots more data out there, and figuring out how we work together to interrogate that data and solve the problems that are coming, I think is going to be essential.